Good day. This is Aljan PD from LO11 class, and for today we'll be having a discussion about the DNA replication. But first, let's have a recap about DNA. So what is DNA? DNA is the short term for deoxyribonucleic acid, a molecule composed of two polynucleotide chains to form double helix. So those two polynucleotide chains are composed of three parts: the phosphate, the sugar, and the nitrogenous bases. And also, DNA contains genetic code of organisms and it carries genetic instructions for development. And in order for that genetic code or genetic materials to be passed, it needs to undergo a process called the DNA replication. So DNA replication refers to the process of creating another strand from the original strand, which means the double-stranded DNA molecule is copied to produce two identical DNA molecules. So where does process occur? So every cell undergo DNA replication, and that process mostly occur at the nucleus, but not in the prokaryotic cells because they have a different region for DNA replication. And in the eukaryotic cells, we have nucleus as genetic information bearer of the cell, or the site of the DNA replication. So when does process occur? DNA replication must occur before the cell division so that the new daughter cell can have the copy of identical or original DNA. So now let's proceed to the importance of DNA replication. First, reproduction of cells and organisms are dependent on DNA replication. Reproduction of cells without DNA replication is very impossible because every cells need that original and authentic DNA molecules, since the information stored in DNA is essential for life. And if a cell dies, the body must replace that cell, and the only way to replace the cell is to first copy the info that the cell contained. Next one is the distribution of the same gene copies. And also, life is independent on the information stored on the DNA. Without replication of the DNA, the traits and info will never be passed on and life would cease to exist. If you see the picture, you can without DNA replication, you can have the traits or the features of your mother or the father. And if DNA replication occurs, you may experience abnormalities which is not that good. And also, DNA replication is the basis of all biological inheritance in all living things. It is the main source of the roots of all inheritance in this world, in the physical world, and all those living things. Let's look at the picture. The mother said, it has your eyes, and which means the baby has had the traits of his father or the mother. Now let's proceed to the stages in DNA replication. But before that, I will first introduce to you the key players in DNA replication. So the key players are enzymes that are very useful in speeding up reaction and other functions since they hold such big rules and responsibility in the process. So first we have the helicase, which is better known as the unzipping enzyme. We'll know about the helicase in the next slides. Next is the primase or the initializer. The next one is the DNA polymerase, which is better known as the builder. And lastly is the ligase or the gluer. So the first stage in replication is the initiation, from the root word initiate, or the act of starting something or the beginning. So here DNA synthesis is initiated at particular points within the DNA strand of origins, but those origins are targeted by the initiator protein, then recruit more protein to aid the replication process. So unwinding of double helix and exposes each of the two strands, and it will utilize as a template for replication. So those unwinding of double helix is the function of the helicase. So just think of a zipper as the double helix and think as a slider as the helicase. So the helicase will destroy the hydrogen band, and it will create an incomplete strand from the original strand. Just look at the picture. That's helicase, how helicase works. So after the unwinding of the first stage of the strand, DNA primer starts to place primers and command the DNA polymerase to start the work. Primers, or the initializers, are synthesized short RNA sequence called primers, and those primers will guide the DNA polymerase or the builders on what to do and where to start. So next stage is the elongation, from the root word elongate, or to expand something. So once the DNA polymerase is attached to the original or to the unzipped two strands of DNA, then the P enzymes will start to synthesize the new DNA to match the templates. But before we discuss the process or the stage more, we have points to remember. First, the DNA strands are antiparallel, which means two strands will not go in the same direction. Since if we're talking to direction, we're not talking about the north, east, south here, but we have in the inner application we have different direction. We have a direction of 5' to 3' and 3' to 5' direction. And the 5', 3' to 5' and 5' to 3' direction. So now let's proceed to the elongation. So basically this is the original DNA strand, the 3 to 5' and 5 to 3'. So as the helicase unwinding, the polymerase starts to build a new strand in 5 to 3 direction. Note the 5 to 3 direction only. 5 to 3, 5 to 3, 5 to 3, creating a new DNA strand which is called leading strand. As we look down, we can see some difference. We can see some gaps. And those gaps, and this strand is called lagging strand. And those gaps are called the Okazaki fragments. So since the DNA polymerase or the builders can works in the 5 to 3 direction, and since this is the original strand is 5 to 3, so the polymerase will work just like this: 5 to 3, 5 to 3, 5 to 3, and also it can only add bases to the 3' ends of the new strand. So that's why this space that where the polymerase stop will be the gap, and it is called the Okazaki fragment. But you don't need to worry about this, because the next enzymes will have the solution for this. So next is the termination. Termination from the word terminate, or the act of ending something. So this is the process of expanding the new DNA strands continues until there is either no more DNA. So the lagging strand will be fixed by the enzymes RNase H and the last key player, which the RNase H removes the RNA primer that is the beginning of each Okazaki fragment, and DNA ligase joins the fragments. That's why it is called gluer. It will join the fragments to create one complete strand, and that's how you create the two DNA identical DNA molecules. Is the ligase. This gap will be fixed by this ligase by joining the fragments. So at the end of the replication process, there are two identical helix molecules. This is one, and we call them semi-conservative because those molecules contain the original or the parent strand, and the newly synthesized complementary strand. Thank you. That's all for today, and thank you for listening.